The sequence of events that convert action potential in a muscle fiber to a contraction is excitation-contraction coupling. In our body, we have skeletal muscles composed of long, multinucleated muscle cells called muscle fibers. Motor neurons coming from the brain or spinal cord attach to the muscle fibers and conduct a large electrical signal called an action potential. The action potential will reach the motor neuron's axon terminal, which stimulates calcium chambers to open, causing calcium ions to flood into the end bulb. As calcium levels increase, small sacs full of acetylcholine called synaptic vesicles undergo exocytosis, which exports acetylcholine out of the cell. As acetylcholine fills the synaptic cleft, it binds to nicotinic acetylcholine receptor. The binding causes ligated ion channels found at the junction that connects the nervous system to the muscular system, or the motor end plate, to open. This causes an influx of sodium ions and an outflux of potassium ions, thus resulting in a end plate potential. If the potential is large enough, it stimulates an opening of voltage-gated ion channels, thus causing muscle action potential. The muscle action potential travels through the cell membrane of the muscle cell called the sarcolemma and into deep vaginations of the sarcolemma called T-tubules. The voltage from the muscle action potential triggers a change of shape in proteins that link T-tubules and a calcium-storing membrane called the sarcoplasmic reticulum. This change of shape allows calcium ions to be released into the sarcoplasm. Released calcium ions diffuse in the sarcoplasm and enter into myofibrils located in the muscle fibers. These myofibrils contain units called sarcomeres. Each sarcomere consists of alternating thick and thin filaments. The thick filaments are myosin and the thin filaments are actin. When calcium coming from a sarcoplasm is present with ATP, an energy-bearing molecule, calcium chemically reacts and bonds to a regulatory protein called troponin. The binding creates a shift in troponin and tropomyosin complex and exposes the myosin binding site on actin. ATP then hydrolyzes to ADP and an inorganic phosphate known as PI. This allows myosin heads to extend and attach to an active binding site on actin, forming a cross bridge. The cross bridges of the myosin filament attach to the actin filament and exert force on them to move. An action called the power stroke is triggered, allowing myosin to pull the actin filament toward the M line, thus shortening the sarcomere. When muscle fibers contract in unison, a muscle can produce enough force to move the body.